DNA, or deoxyribonucleic acid, is called the molecule of heredity and is located in the nucleus of a cell. It is called this because it passes down traits from generation to generation and is the genetic blueprint for life. DNA is in the shape of a double helix moving anti-parallel and is made up of nucleotides. Nucleotides are made up of a phosphate, deoxyribose sugar, and a nitrogenous base. The nitrogenous bases are adenine, thymine, cytosine, and guanine. The nitrogen bases adenine and thymine are always connected together by 2 hydrogen bonds, while cytosine and guanine are always paired together with 3. Proteins are needed for all organisms to function, and these essential proteins are created through a process called protein synthesis.
The first process in protein synthesis is the copying of the DNA to the RNA through a process called transcription. First, an enzyme called RNA polymerase unwinds the double helix with the help of helicase, then reads one side of the helix called the promoter. It makes an exact copy of the side, with one exception. It replaces all bases that would be thymine with something called uracil. What is produced is something called pre-messenger RNA. Then it undergoes some editing, where it gets something called a methyl cap at one end, as well as a poly A tail on the other end. Parts of the strand called introns are removed from the strand, and the remaining exons are spliced together. Once these things are added on, it is a completed messenger RNA strand, and it leaves the nucleus into the cytoplasm.
Once in the cytoplasm, a process called translation begins. Translation is when the ribosome translates the mRNA into amino acids and peptides. tRNA helps the ribosome function by bringing in the anticodons. The ribosome reads RNA by dividing it into sections of 3 bases called codons. The anticodon is a series of 3 bases that are complementary to the mRNA. For example, if the codon was AAA, or 3 adenines, then the anticodon would be UUU, or 3 uracils. The ribosome is a large complex of proteins and ribosomal RNA, rRNA. The ribosome consists of many small subunits that bind on the mRNA to produce polypeptides, which are a chain of multiple amino acids. At one time, multiple RNA can attach to one mRNA and synthesize at the same time.
Once the amino acids are assembled to form the polypeptide, the final step of protein synthesis begins. There are 4 main levels or structures of this step. The first, the primary structure, is simply the order of the amino acids in the polypeptide chain. The secondary structure consists of the alpha helices and the beta-pleated sheets. The alpha helices are created by the coiling of the chains, and the beta-pleated sheets are created by the folding of them. The next structure is the tertiary structure. It is made up of the folding of the beta-pleated sheets and the alpha helices upon each other to form a 3D structure. Finally, there is the quaternary structure. This is formed when there are multiple different chains of amino acids that fold upon each other. Once all this is completed, you are left with the protein, which will go and help your body.